My name is Stephen Nowell. It is 5/15/2022. This is Unit 4, Case Connections. So I did case 1, and in this case, a newborn was born and shortly after began developing lethargy, hypothermia, and apnea. Laboratory workup was completed in order to diagnose this patient, and results showed an elevated ammonia level at 314, which then rose to 600. In this study, we will discuss the underlying mechanisms of ammonia disposal, the urea cycle, and why this newborn is experiencing dysfunction of her ability to excrete excess ammonia. To begin, it is important to have a solid background on how nitrogen disposal occurs in a normal setting. Amino acids which contain nitrogen are integral to numerous body processes such as protein synthesis, fatty acid synthesis, and gluconeogenesis. Although these amino acids are important for their roles in the body, once metabolized, the amino group must be disposed of due to the harmful effects of excess ammonia that accumulates from metabolism. The main mechanism for nitrogen disposal in the body is the urea cycle, which occurs in the liver. In order to undergo the transformation of ammonia to urea, the ammonia must first be transported from extrahepatic tissues to the liver. The first major pathway in tissues involves glutamate being converted to glutamine through the enzyme glutamine synthetase. Glutamine is one of two main non-toxic transporters of nitrogen and will help transport ammonia to our hepatic cells. Once in the liver, glutaminase will act upon the glutamine and convert it back to ammonia and glutamate. This freed ammonia is now in the liver and ready to enter the urea cycle for disposal. Following, glutamate can be further converted to alpha-ketoglutarate through glutamine dehydrogenase. This reaction helps release additional ammonia and also allows the remaining alpha-ketoglutarate the ability to enter the TCA cycle, where it can be utilized for its TCA cycle intermediates. Of note, this reaction is known as a deamination reaction and is the reverse of transamination. This reversibility is extremely useful as it allows ammonia to be fixed for use in biological pathways or freed to enter the urea cycle as we have described above. To clarify, transamination reactions are performed by enzymes known as transaminases and are used to remove nitrogen through the conversion of an amino acid to a keto acid, while simultaneously resulting in the formation of a new amino acid and a new keto acid. A good example of this occurs in muscle cells when glutamate, in conjunction with pyruvate, will be transaminated to alanine, which is our second major carrier of excess nitrogen to the liver. Once in the liver, an additional transamination occurs in which alanine releases its carbon skeleton for use in gluconeogenesis, while also releasing the nitrogen that will then enter the urea cycle. This reformation of pyruvate from alanine is known as the alanine-glucose cycle and is one connection between nitrogen transport and gluconeogenesis, as pyruvate can be used as a substrate for gluconeogenesis. Once ammonia is in the hepatic cells, the urea cycle will take place. Of note, this cycle utilizes a feed-forward mechanism, meaning the more amino acids that are degraded, the more this cycle will be performed. The first step incorporates ammonia, bicarbonate, and 2-ATP, which are then converted by carbamyl phosphate synthetase 1 into carbamyl phosphate. Next, our carbamyl phosphate will combine with ornithine via the ornithine transcarbamylase to form citrulline. Citrulline will react with aspartate to form arginosuccinate through arginosuccinate synthetase. Arginosuccinate is then cleaved via the enzyme arginosuccinate lyase, which results in fumarate and arginine. Fumarate will convert to oxaloacetate, which is then used to provide TCA cycle intermediates, while arginine will be broken down to our end product of urea, which will then be excreted, and ornithine, which will be recycled for use in this cycle. Of note, the metabolism of amino acids occurs both in the fed and fasted state, in the fed state through dietary means, and in the fasted state through cortisol-mediated protein catabolism. Thus, the urea cycle and these processes will be ongoing. With a solid understanding of the normal physiological pathways and reactions, let's now look at what went wrong with our newborn patient. The further laboratory analysis for this patient revealed an elevated urine orotic acid level and normal plasma citrulline level. Orotate, also known here as orotic acid, is an intermediate present in pyrimidine synthesis, and excess orotic acid will indicate dysfunction of the urea cycle. Pyrimidine synthesis also uses carbamyl phosphate in pursuit of creating the pyrimidines thymine, cytosine, and uracil. If there is dysfunction of the urea cycle in which carbamyl phosphate is in excess, then the excess product will enter the pyrimidine synthesis pathway. The elevation of urine uric acid is how we identify this buildup of excess carbamyl phosphate. In the urea cycle, we now know that carbamyl phosphate synthetase 1 could not be our deficient enzyme, as we have an excess of carbamyl phosphate, which is produced by this enzyme. The low citrulline level also indicates citrulline is not being adequately produced in our urea cycle. The only enzyme between citrulline and carbamyl phosphate is ornithine transcarbamylase, also known as OTC. Actually, OTC deficiency is not surprising, as this is the most common urea cycle deficiency and often manifests with neurological issues, as we see with the symptoms present here. In cases studied where patients had OTC deficiency, it was found that arginine became an essential amino acid. This occurs because arginine is found after the defect, so similar to how citrulline will be low, it will also be low. The other amino acid present in the urea cycle is aspartate. However, aspartate would not become an essential amino acid, as it is synthesized by glutamate to aspartate in other areas of the body. In conclusion, this patient is most likely experiencing ornithine transcarbamylase deficiency, as evidenced by increased ammonia level, increased urine uric acid level, and decreased citrulline levels. Treatment for this patient would involve extensive workup and care in a hospital by a team of experts, and care would be taken to reduce the ammonia levels through medications, potentially renal replacement therapy, also known as dialysis, and this patient may eventually need a liver transplant as well. The next case is case 2, which provides 3 scenarios. So this case presents us with 3 scenarios in which there is an enzymatic deficiency that is affecting amino acid metabolism. In the first case, a seemingly healthy newborn was released 3 days post-birth with minimal complications. However, newborn screening revealed positive branched-chain amino acids. The branched-chain amino acids are isoleucine, leucine, and valine. The role of these amino acids is to serve as an energy source for proteins, primarily for skeletal muscle cells, and to help replenish the intermediates of the TCA cycle through carbon donation. In order to use these amino acids, they must be broken down. The first step in this degradation is to be transaminated in order to remove nitrogen from the amino group. Next, the resulting keto acid from transamination will undergo oxidative decarboxylation by a branched-chain alpha-keto acid dehydrogenase complex. This reaction produces NADH and FADH2, which can be used for energy creation. Additionally, valine, isoleucine, and leucine will produce succinyl-CoA, acetyl-CoA, and acetoacetate respectively, which again are our TCA cycle intermediates. In this scenario, the presence of elevated branched-chain amino acids means this degradation is not occurring, and these amino acids are building up. So these are our amino acids that are not getting metabolized and are the most likely culprit for this deficiency is our branched-chain alpha-keto acid dehydrogenase. Although the hallmark sign of burnt maple syrup is not present, the elevated branched-chain amino acids and lack of familial genetic disease would lead us to believe this patient is experiencing maple syrup urine disease. MSUD is an autosomal recessive disorder, meaning the parents could both be carriers of a recessive allele for this deficiency, where no one in the family had gotten the recessive trait up to this point, and yet this patient still can get this disease. Moreover, not all newborn diagnoses of MSUD present with symptoms, which is additionally consistent in this case. There are no amino acids downstream of our branched-chain amino acids, so no additional amino acids would become essential from this disorder. The treatment for MSUD would be to limit dietary intake, which would mostly be protein restriction of our branched-chain amino acids. Moreover, the branched-chain alpha-keto acid dehydrogenase complex is similar to our pyruvate dehydrogenase complex in many ways, and it also requires thiamine as a cofactor. So these patients would also take thiamine supplementation. In our second scenario, we find ourselves with a 69-year-old woman with a history of chronic back pain of the hips, shoulders, and aortic stenosis, who presents with blue-gray pigmented macules over the cartilaginous portions of her eyes and ears. The symptoms presented are most likely due to acrinosis, which manifests as early pigmentation changes of the ears and sclera. Additionally, one can develop acrinotic arthritis, which can commonly affect the lower back and other large joints. The underlying disease that causes acrinosis, alkaptonuria, may also cause cardiovascular disease. Our patient's macules, chronic back pain, and aortic stenosis perfectly fit the bill of this diagnosis of alkaptonuria. So let's talk about it from a biochemical perspective. Phenylalanine is a ketogenic amino acid which can be used to produce fumarate in the ketone body acetoacetate. In order to get these outcomes, phenylalanine is converted to tyrosine, which then undergoes a series of reactions to eventually form the desired acetoacetate and fumarate. Alkaptonuria is characterized by the deficiency of hemogentisate oxidase, which leads to the accumulation of hemogentisate. The excess hemogentistic acid auto-oxidizes and can result in all of the symptoms described above. Although tyrosine is derived from phenylalanine in the process described above, the deficiency of the hemogentisate occurs after tyrosine has been created, so no amino acids will be deficient with this disorder. Treatment for this deficiency is mostly symptom management. There is ongoing research regarding nitisinone and ascorbic acid, which may help reduce homogentistic acid. However, the results and efficacy of this treatment are not yet widely agreed upon. In our final scenario, we are presented with a 4-day-old infant post-birth with no complications. It is noted in lab work that the phenylalanine level is elevated at 6.6. Given the information discussed in the previous scenarios, we can reasonably assume that this deficiency is additionally related to phenylalanine metabolism, as that level is high in our patient. The first step of this breakdown in pursuit of acetoacetate and fumarate revolves around the conversion of phenylalanine to tyrosine via the enzyme phenylalanine hydroxylase. An important note about phenylalanine hydroxylase is that it requires the cofactor tetrahydrobiopterin, also known as BH4, for its hydroxylation reaction. Since we know via the lab work that phenylalanine is not being metabolized to tyrosine, we can conclude there must be a deficiency in our enzyme phenylalanine hydroxylase or a deficiency of the cofactor BH4. And the disorder associated with both of these things is known as phenylketonuria, also known as PKU. So PKU screening occurs at birth, as PKU can cause neurological deficits, tremors, seizures, and hyperactivity. The treatment will be limiting ingestion of foods that contain phenylalanine, depending on the causative agent. Tyrosine supplementation may or may not be necessary. In the event that our enzyme is deficient, then supplementation of tyrosine must occur, as it will become an essential amino acid. However, if BH4 is deficient, dietary supplementation of the cofactor has been shown to help reduce the phenylalaninemia. So again, this was Unit 4 Case Connections. Thank you.